Hello, everybody. Welcome to class on learning how to plot our data. We've started to generate a data frame of number of overlaps with promoters and gene bodies, and we're going to learn how to start to plot that data, focusing on GGPLOT. So we're going to go through the basics of GGPLOT in this class, and then this next plotting class will take our data and focus in on those skills that we'll use moving forward. Okay, so let's get started. We're here in class 2023, and we are in 05, our analyses plotting. We've generated our data, and now we got to see what these plots look like. How are the trend lines looking, etc. And so this is the code we're working on today, 01 plotting.rmd. And the goal here, again, I'm going to use the code from my RStudio, but if you're taking it from our website on long-non-coding-rna.io/teaching, then we are in here in ChIP-seq analyses plotting our data. Okay, and so here we are with the code getting set up. We're going to need to load ggplot, and ggpubr is super useful. You don't necessarily need it, but you're going to want it for plotting regression line values and other things. And I've cloned in for you some plotting functions. We're not going to really use them in this class, but just so you know, in your util, there's that. And in this underscore setup in the util, are some themes that we'll see that you can set the kind of theme you want your figure to look like and pick from different ones. And we have a custom one that's in this setup file. So I'm just going to source those, even though we're not going to really go over them. Our main goal today is just to go over the basics of ggplot, or the grammar of graphics. It's amazing. You can basically build your own figures. And we want to apply learning these skills to our data and plot, number of peaks versus number of mRNA promoters and link RNA promoters and other basic comparisons that we'll want to do, histograms, densitygrams, that kind of thing. So to do that, we would load our data from our previous class. I'm here again in our analyses, plotting 02 and plotting 1. We previously in 01 at peak features in our results made a numpeaks df here, and we're going to load that in and take a look at it. I've set my working directory. I'm going to go back to it. So plotting, okay, read this in, and we see our numpeaks df that we made in the last class with all these different values we could plot against each other to see if there's trends or which DNA binding proteins don't follow trends, etc. And because we only have 6 DNA binding proteins for this version, so we're learning in layers here. We're going to go through with just our custom ChIP-seq data set, but then we're going to move to 480 DNA binding proteins. And so we're going to have a sneak peek glimpse at that data, what you're going to generate in the next iteration of the class. We'll take these skills and then apply it to a bigger data set. And we'll sort of hone in on more and more on getting these functions done more succinctly. But for now, going over all the basics. And so let's read in that bigger data frame. You'll see here looks very similar. We'll call it big numpeaks. And we can see it just has 460 rows, so 460 different DNA binding proteins, has that information we read in about the TF and DNA binding domain, overlapping mRNAs, link RNA promoters, et cetera, et cetera. So we're going to take a look at that data. It's actually from last year's class, and it can be downloaded here if you're not on class, in class with us. You'll need this data frame for today. Okay, and you can go back and run it on your own data frame, numpeaks df, just by changing the areas we have big nums to your numpeaks df. Okay, so we're going to start with ggplot. The first layer here are outlined, and there's more information as we go, is the data line. Where is the data? And so what ggplot wants first is where the data is. So we're going to give it numpeaks df. And we're going to say AES is something to remember for any ggplot, is we're setting the aesthetics of the data layer. And so this is the data, and this is the aesthetics of that data, which is the x-axis is equal to the number of peaks. So in our data frame here, we have number of peaks is the second column. So it's going to take that as the x-axis, those values, and the y-axis will be total peak length. And we can compare those two. So this is a core part of any plot. And then anytime there's a plus, we're adding another layer. And this is really great because you can customize your figures to just the way you want them. And we're going to do a point plot for now. We'll do bar plots, histograms, and other things as we go. But we'll start with just a simple point plot. Okay, so we have our big num peaks df loaded in. We're going to do num peaks versus total peak length. Okay, all right, so we got a plot already. And what we can see here, if we zoom out on it, that there's a linear trend that the more peaks you have, the more DNA and the genome you cover. And that kind of makes sense that the more peaks you have, the more sort of DNA you would think is covered. However, some DNA binding proteins might have small peaks. And so having many peaks might not be that much more. So we don't know what it is, but we do know that it's correlated, that the number of peaks is correlated linearly with the amount of total DNA covered by those peaks. And this is the same thing. A preferred way I do is tidyverse, where we just pipe in the data frame with this pipe here, and then do the same thing with ggplot, set the aesthetics, and then with a point, and it's really going to give us the exact same figure. Okay, so now let's say we want to change the shape of the points. So we're going to go into this layer here, in the aesthetics layer is where we are. Sorry, we've set the aesthetics, and now we're going to go into g_on_point, and we just left it blank before. Now we're going to add parameters in there. And we're going to say make the shape a square and the color purple. And so all of a sudden we can see we have purple squares, the same data. Okay, so that's really nice. And you can look here for a bunch more shapes and colors of your interest that you can set your figure to your setting or your favorite settings. Okay, so now we're going to get a little deeper in here in the aesthetics layer, and we're going to use color, which is going to be really handy, that we can color this data two different ways, whether it's a transcription factor or not, or what type of DNA binding domain it has. And so what we're going to do here is we have a DBD column in our big nums peak here, DBD here. And so we can pick out a type of that and color those and see, are they along the linear trend or do they deviate from that trend? And so this is the same aesthetics we've set up here. And now just in color, we're going to set it to the DBD column in this data frame. And it has to equal this type of DNA binding domain. Okay. And so then now what we can see is we get true, false, and NA. So it's true if it has this domain, it's false if it doesn't, and NA means it can't tell. And so what we can see here is the trues and falses follow the same pattern. And so we can kind of conclude that the zinc fingers don't have more or less peaks. They kind of follow the distribution of the other DNA binding domains, and that they both sort of follow the same linear slope there. Okay, so that's nice that you can kind of just dig in here as the main point and add a color by this, by a factor you're interested in the data frame, in the aesthetics layer. Okay, so now we can actually add functions in the aesthetic layers. So let's say you have big numbers or you want to log to them. We can set up the aesthetic layer here to now have num peaks like we did before, but divide it by 1,000. So the number of peaks per 1,000. and Y is going to be the total peak length divided by 1,000,000 to make this a little more scale, a little more readable. And we're going to do the same color by DBD equals this and a geom point. Okay, wow. Okay, so that changes things. So you got to be careful on what type of scale you have. So putting this in log 2 shows us that as the number of peaks goes, that the amount of DNA starts to increase as you have more and more DNA binding peaks. But we just saw a linear trend, and that makes more sense. And both of the zinc fingers are following the same trend. Okay, so that's just to show that in the aesthetics, you can run a function on your data and manipulate it there. So it's on a different sort of scale and can give you a different perspective. Okay, the facet is another amazing layer to go here, is that we have a bunch of different categories and get different columns, like multiple different DNA binding domains. What if we wanted a figure for each one of them separately? Well, facet is the thing to do that. So you can take a column and all the different variables in that column, you can separate them out and make their own figure in them. And so let's step through that here, where we're setting up the aesthetics same as we have, number of peaks versus total peak length. And now we're going to add this facet wrap here. And our TF column has two categories, yes and no, whether it is a TF or not. And we're saying take the TF column and facet it in this facet wrap layer, which we added with another plus here after the aesthetic layer, and do facet wrap function by the TF column and do that here, and then plot the points for them. And so we're going to get 3 plots here, one for yes, it is a TF, and we can see that the more number of peaks it has, the more DNA space it covers and is linear. Same for yes, and then NA as well. So in all of these cases, we can see that if you're a TF or not, or we don't even know, that the peak properties are linearly correlated with number of peaks versus how much DNA is covered. So we're learning a lot. We're digging in and seeing that pretty clearly, no matter how we cut it, number of peaks corresponds to the amount of DNA covered. Okay, so now we're going to try a different geom histogram. We've been doing geom point. So we can now think about our aesthetics is a bit different with the histogram, because our y-axis is going to be the frequency of observing that event. And so the x-axis will be the number of peaks, and we'll see what the distribution of the number of peaks is, or how often those numbers occur, using the histogram, where we're adding it with the plus again and doing geom histogram. So we can run that. Excellent. So now what we can see is we have peaks ranging from 0 to 1.5 million, or sorry, 150,000, 50,000, 100,000. So most of them are before 50,000 in here. And so that's kind of nice. We can see that most of our data is here, but there's a distribution that goes all the way out to here. So that might be something you want to present at lab meeting, the variation in the number of peaks your samples had. So now what we're going to do here is just add the number of bins to 30. So we can go into this geom histogram that was blank and add more bins to give us more resolution. And I encourage you to pause and sort of look at that, play with this a little bit, where you can add sort of like 300 and get it sort of at more of a fine grained resolution, where we can see actually most of the peaks are over here below 25,000 and so on. So, okay, so that's a histogram. And we can still do histograms with filling by colors. And so this is the same aesthetics, x equals number of peaks, but we're adding a comma to put another parameter in here, which is fill equals the TF. So we're going to say whether it's yes, no, or NA, and plot the exact same thing in big nums peak DF. And so what we can see here is that the distribution of the number of peaks is pretty similar between whether you are a transcription factor or not. So transcription factors don't seem to have a smaller number of peaks, is another observation we're just making now. And we'll go over this again and record it more formally. We're just learning the basics of ggplot. So we can add this parameter in the geom histogram of Dodge, and that will separate these distributions here for us. And we can see them separately. It's not the most pretty thing. So we can also look at this as a density plot, as a kind of similar way of thinking about it, is where is most of our data on the x-axis? How much of the percentage of the data is at a given number of peaks, and we're going to do fill as TF again, and this time use geom density instead of histogram. And so what we see here is the distributions of whether you are, since we did fill by TF, we got these 3 different plots, whether you are not, whether you are, or whether NA, that your distribution of the number of peaks here. And so we can see in most cases, most of the density is here. The area under this curve would equal 1. And so you're seeing where the density of the data is. And for all 3 distributions, it seems to be over here to the left. So it's kind of a different way of looking at a histogram. I prefer density plots often. So here is the same thing. Number of peaks is X, and fill by TF, and we're going to turn alpha on in our density thing here. So you can see each time we add a different geom, we can go in and find some of the parameters, and you can find those in the manual as well. And so there we just turn the alpha to 10%, so we can now see the distributions all having a similar shape, and they're sort of all put onto the same scale, whether they have 400 representatives or 200 or 100. Here we're doing the same thing, fill by TF and color by TF, and have the density at 0.3 just to play with these parameters. And so now these are colored by the box color as well. And then finally, if you don't want to see any of that color, you can turn the alpha to 0 here and just get the lines plotted by the colors of whether they are or not a TF. Okay, so we're starting to learn things while learning plotting, hopefully. And now we are going to see how you can do multiple geoms. So we did just like geom point, geom histogram, geom density, and now we're just going to play around just to sort of learn the basics here, where we're going to have the same setup of putting in the data frame, num peaks, and y is total peak length, that linear correlation. We're going to do a geom point and then also a density to see where most of the data is. So we're kind of combining the two. And we're just going to leave the parameters blank for now. And each time we add a layer, we add a plus. And this is our final layer, so it doesn't have a plus. And we go from there. And voila, what we can see is our same linear trend we saw before. But now we can see that most of the density of the data is here. You can think of this like a topological map of climbing up a mountain to the summit here. And so most of the data is down here. And so it gives you not only the linear trend, but where most of the data is coming from. The order kind of matters. So we did point and then 2D. And you can see the lines are on top here. And if you wanted that switched, you could just switch the order. That's just to show that the order matters, and the last argument is going to be on top here. Okay, so let's say we wanted to add a trend line. We kind of see a linear relationship, but let's formalize that and add a trend line to it. And we can also have the density there as well. And so we can look up geom abline, where you can give a slope and intercept of what you predict your data is, and then plot it and see if it's on or off that line. Or geom smooth is a function that can use like a linear model and see if there's a linear pattern in your data. And that's what we'll do here by adding a geom smooth line here. Okay, so we're doing the same setup, num peaks versus total peak length, what we have here. We're adding a geom point, a geom ab line where the slope is 1000 and the intercept is 0. So we're just making this as a straight line that then every 1000 peaks, you would see a linear increase in the number of DNA binding or total DNA covered. And we are going to use this geom smooth LM, which is really handy. This is probably what you're really looking for. That will put a smooth line in it. So if we ran it without that, we're going to get our custom line. Oops, except for I need a parenthesis. Okay, so now I just put that line in there. So it's true that for every one number of peak, you gain about 1000 base pairs of DNA, or this is my guesstimate. That looks pretty much correct. And then we can add the geom smooth, which will do a linear model. And you can look at the different type of methods. There's GAM for general additive models and other types, but a linear model is probably what you're most used to and seeing. And so now we can see there's some gray here showing some variance, but there's a really smooth linear trend line that explains this data almost as good as that 1000 peak line. Okay, so now let's go to another geom, bar plot. Common thing to want to do. So we can put in our data frame and set the axis in a bar plot. Again, the y-axis is going to be set for us with the number of events, and we'll do it with DBD as the DNA type of DNA binding domain the DNA binding protein has. Don't say that too fast. Okay, so we can do that, and we can see we have all these different DNA binding types, and the C2 H2 zinc finger hook domain has the most entries, so maybe that's one we want to look at and play with. The rest kind of have a similar distribution along there. Okay, so so back to here. So what we're going to do is that's kind of why we picked that Z2 C2 H2 before. And so we can now see who's most represented in our data. Maybe a good plot to show. And then we're going to take here and do X is the number of DBDs, and we're going to now make the Y the number of peaks. So we're going to see how many peaks there are relative to how many representatives there are. And to do that, we need to go into geom bar and say statistic equals identity. And that is going to mean that we're going to take the identity of the y-axis and plot that. You shouldn't need to do this very often, but I just thought I'd use this as an example. And so now we can see for every DNA binding protein, the number of peaks is plotted as the y-axis. And so that there we can make our DBP is the x-axis here, our 460 DNA binding proteins and the number of peaks. And you can see this is really messy. Even when you zoom in, it's hard to read and isn't really telling us much. So that's why usually when a bar plot, you have a variable that a category you want to plot the numbers in, and so that you would just use this up here as a much more common way of not going in and doing that. It's just to show that you can do a lot of things inside these different geom layers. Okay, so now as a more reasonable thing to do here, we're going to say the x-axis is whether you are a TF or not, and y is the total peak length, the amount of DNA you have. We'll make that as a geom point and do a statistics summary where we're going to put function is equal to mean, color is red, the shape of that is 2, and size is 2. And you can play with these, and you don't have to have the shape be 2 as a triangle. You can change it to 3 and see what shapes you can get. But we're doing a statistics layer here where we're doing a stat summary on the data in here in this total peak length value of y. And so we can put the mean on there. Let's take a look at it. And so here we can see what the total peak length is if you're not a TF, if you are, and if you don't know, and we can see they're about the same means, meaning that it doesn't really matter how many peaks you have, doesn't matter if you're a transcription factor or not. So that's an interesting finding, that there's no bias towards a certain category. Okay, and then we can use a linear model also to fit a trend line with this, which we did up above, and really that is redundant. So what we've gone over so far is the data layer. What is the data? What is the x-axis, y-axis if you need it? How to map that data, in the aesthetics layer, or mapping. Statistics, where we took the mean of the data inside a stat layer that we called up, and geom, the different types, points, histogram, density plot. And so we're going to sort of fine tune some of these things, and then in the next class sort of go focus on the ones we really want to use. But just to kind of cover all the layers here, the scale layer is important. Some things you want bigger or smaller in your plot. And this is why GGPLOT's great, is that you can change variables on every single layer here. So we're going to put in our data frame and set the x-axis is to the log 10 of the number of peaks. Y is going to be the log 10 of the total peak length. We're going to color by TF. So this is all the same stuff we've done before. Make a geom point, and we're going to scale x continuous as a scale layer function to have breaks only at these 4 places. And I'll run this before we add that next piece on there. And so what we can see here is that the scale x continuous said, I only want to go up to 4. And so you can tell it how to scale this. And we can see, again, this linear trend between the number of peaks you have and the total DNA you bind. And you can change the colors of these things by, you can Google these as well, different palettes that you can load in here on the scale section. And let's add that in. And it just changed the color of the dots to a specific pattern. And you can figure out your favorite palette, and then moving forward, you're sort of done and just add that in. Okay, so now we might want to, we've been messing with the scales, we might want to actually set the limits of a image. And here we might want to go to 1e to the 5th out here to fill that in. And so we're setting up the same thing, number of peaks, total peak length, and we're going to do a geom point. And if we looked at that without any of these limit changes, what we would see is that, well, a lot of stuff out here, there's not much out here. So let's hone in more on here, around 100,000, less than 100,000. Okay, so that's 1e to the 3rd. And so we're going to start somewhere over here and go to here, where most of our data is. We're kind of just zooming in. And so now we can run that with that on there. And what we can see now is the linear trend sort of zoomed in without that other data on the far end. So you can just set your X limbs as a new layer in your image here with X limb and Y limb, and we'll use that quite often. So we saw breaks with histogram. You can add more breaks, and you can easily just set your X limb and Y limb as those. So here's where one of the best things about ggplot is faceting. So you can make take this figure and make it for every type of DNA binding domain there is, instead of just whether it's a TF or not. It's only 2 categories, and I think there's like 17 categories. So we get 17 different plots. So you'd see every individual example plotted against itself. And so let's look at that, where we might want to just look at these 3 types of DNA binding domains. The same one we've been looking at, we'll add homeodomain and bzip. And if we want to just look at what these are again, we're going into big num peaks DBD, the type of DNA binding domain column, and we're indexing again with the square brackets. We're not subsecting, we're indexing and we're ordering the table command to be in the order of the DBD names. So we can see that here. And our result here is that they're in order from lowest to highest number of representations here. And we can see this one is one of the biggest ones. And so we'll take that. Homeodomain only has 3. And so these are the categories we can grab from, and we'll make an object for the 3 we're interested in here, just using the concatenate, and we'll call it DNA binding domains as the object. Okay, so now what we can do is only plot these types of DNA binding domains. So here we're doing GGPLOT, where we're going tidyverse and putting the data frame into a pipe. I probably should have it like this. Then we're doing a filter on the DBD column in our data frame and the ones we want here. So we're just taking these out basically. And then we're setting the x-axis to num peaks, total number of peaks, and we're doing a facet grid here where we're saying, okay, what we did that with TF above, and we're going to say take the DNA binding type and facet it here with the dot meaning here. So we're going to get 3. These are the 3 things it's going to facet on in DBD. Okay, so and then add a point there. So if we do that, what we see now is what type of domain you are is what we separated on, and the type of DNA binding domain. And the only thing that was piped down this far were these 3 because they were sub-selected here with this DBD in DBDs. So this is just an example of showing you how you can, inside these parentheses, send information down. It's almost like you can code in GGPLOT and have it customize your figure here. So we see basically the same trends, that whether you're any one of these 3 types of DNA binding domains, there's a linear trend there. And we could add that linear smooth model on here just by adding another plus, et cetera, et cetera. Okay, so you could also facet 2 parameters inside of this figure here. So we could add another figure inside the figure, if you will. So here what we're doing is we're using, again, more of a tidy way of doing this, showing you both ways. You can take your preference. I prefer this way, where you pipe in the data frame. So now it knows all the column names. And so it's going to filter DBD column in this data frame that it knows everything about now by this DBD's object, which is those 3 we picked. The x-axis is going to be the number of peaks, total peak length, same as we've been doing. And now we're going to facet the grid by the DNA binding and whether or not it is a TF or not. So we're faceting the grid into more categories, and we're going to get more plots because we're saying for each type of DBD, we have 3. Tell me whether it is a TF or not. And those answers we know are yes, no, and NA. So if we take a look at what we get here, what we can see is, I guess we just got yes and no. Oh, because I picked 3 that don't have NAs. That's why. So the beta zip we see has the same trend, and it doesn't matter whether you are or not a transcription factor. In fact, most of these are transcription factors because we're in the yes category versus the no. So again, I know this is a lot to take in, but it's kind of cool that you can just facet and then split it into different variables, and you'll get each of those plots individually. And is a very handy way to make genomic plots. So then we could do the same thing here, where we're just switching DBD from DBD to TF to TF to DBD, and the facet order sort of matters. It's just going to change the order now, where no is the row, no and yes is the rows, and these are the columns. So this is going to be the rows, and this will be the columns. Okay. Excellent. So now we are have done facet wrap. And if we want to get deeper into those facet wraps, we can also customize this to the number of rows, number of columns. So we can do this, and change it to that, or we could now make it a facet with only 2 rows here, and we're going to get an odd third one over there. So you can go over that and just play with some of these things. And scales free is worth noting here. So in facet wrap, we previously just did this. Now you can see that adding a comma, you can add more parameters, such as scales free is a good one to know. That is going to, instead of forcing the scales to be the same, it'll normalize it to each column. And we can say the number of columns is 1 and plot that. And we can see now they all are going to have a little bit different of a scales because of scales free. The coordinate layer is something you probably will never touch, but it is the last layer next to theme. And so it's just worth doing really quick due diligence on this, where we've made this plot before, where we plot the number of instances of a given DNA binding domain here with C2H2 zinc fingers being the highest. We can also add another, put this in a different coordinate system, which would be polar, which is like a circle. Again, you're probably not going to know this, but just so you know that there's another layer of coordinate that starts with coordinate and then polar, and there's many different coordinate layers that you can easily look up. But then this just puts it in a circle, and it gives us the values of each one as like how far out. So most of them have very few, but this C2H2 really sticks out. Just a different way of plotting the same thing. And you can, in the coordinate polar, look up different ways of changing the axes, and you can change this to have it only do a quarter circle. Again, not something you're probably going to ever get out of Cartesian courts, but in due diligence, that is how flexible GGPLOT is, in that it allows you to do functions, customize, scale, add other features, wrap, and then split that into a bunch of figures. So it's a really great way to make figures, in my opinion. And here is themes, where we set a theme in our R setup that we loaded, and it's here paper white. So in our setup function, we have this theme paper white, which gives us a specific size and things we want about our figure. And so here we're just plotting the density plot of the number of peaks for given DBPs with an alpha. We're specifying the colors. You can look up your favorite colors, but they all have a number. And so you can put in the number and it will make it that color. And now we're adding a theme layer of paper white, which has our dimensions and other features here, where we're telling things how we want it to look. And it's actually quite extensive. It has other parameters we can use, and as a lab now we can all use the same theme and make our own little tweaks. Okay, and then this is just adding titles you can add with X label, Y label. You can put the title. And so here, if we do this, what we can see is mRNA and link RNA genes, the number of binding, promoter binding events here in our data frame, and we're going to do with our data in the next class. The density is here, what we've seen before. This might be a nice figure that you might want to plot and put in a paper, etc. There's numbers of themes you can get, and you can sort of Google them, but there's like theme Cleveland instead of paper white, same plot in theme paper white with a geom density. So, all right, well, that is our full coverage of ggplot. And now we're going to move on to take our custom ChIP-seq data sets and get out of them what we can, and sort of use these basic skill sets again, reiterate them and apply them to different aspects of our current ChIP-seq data. And this was more of just a total overview and a sneak peek at our bigger data set. All right, so with that, thank you and be well.